Hey guys, welcome back to CCL Biology, the channel for easy to understand biology lessons. If you're looking for help with general, AP, or college introductory biology, this is the place for you. I am your friendly bio nerd. Thank you so much for tuning into my channel. In today's video, I'm going to be talking all about carbs, carbohydrates that is. So if you want to get a better understanding of carbs, then stay tuned. While you're here, don't forget to hit the like button and comment below if you find this video to be helpful. And if you want more easy to understand biology videos, then be sure to subscribe to my channel and click the notification bell to get updates about new videos as soon as they come out. This is part 2 of a 2-part video on carbohydrates. In part 1, I talked about the structure of simple carbs, so be sure to check that video out if you haven't already. I'll put the link in the description box below. In this video, we're going to look at the structure and function of more complex carbohydrates. A carbohydrate is an organic compound and one of the 4 macromolecules that serve as the building blocks of organisms. Carbohydrates, or carbs, are sugars. They can be simple sugars, like the ones I talked about in the part 1 video, or complex sugars, like we'll see here. The complex sugars are called polysaccharides, which literally means many sugars. Although poly, that prefix, is technically anything greater than 2, polysaccharides usually consist of hundreds or thousands of sugar molecules that are joined together by glycosidic linkages through dehydration reactions. Now that's kind of a lot, so let's take a closer look. So here we've got 2 monosaccharides, or single sugars. The monosaccharides use a chemical reaction called dehydration synthesis, and that bonds the 2 monosaccharides together to make a disaccharide. So just to follow along with the picture here, we've got this monosaccharide, which happens to be glucose, this monosaccharide, which also happens to be glucose. So they come together, and the reaction that brings them together is called a dehydration synthesis reaction. So once they come together, it's then called a disaccharide, or 2 sugars, and the name has changed. This is called maltose. So these 2 glucose molecules have come together, and they're bonded here with a covalent bond. Now this particular covalent bond in between these monosaccharides is called a glycosidic linkage. So be sure to check out my video on macromolecules to learn more about dehydration synthesis reactions that join monomers together to build polymers. So there would be another dehydration reaction each time a new monosaccharide is added to this chain, and eventually it would grow into a polysaccharide. So that's kind of what we're seeing here in this illustration. So this is an illustration of the polysaccharide, which is starch, which we'll talk about in just a little bit. And here's a little cutout showing 3 monomers that are linked together. So in order for these 2 to come together, there was a dehydration synthesis reaction and then another dehydration synthesis. So for each of these little hexagons that you see here, there was a dehydration synthesis reaction that had to join each hexagon to the next. Now, what's the point of joining a whole bunch of sugar molecules together like this? What do polysaccharides do? They can do one of 2 things. They can store, or they can build. Polysaccharides can store energy, or they can be used as building material. Now, there are lots of different polysaccharides out there, so how do we know which polysaccharide will do what? How a polysaccharide is built, aka its structure, and what it does, aka its function, all depend on the type of monosaccharides that the chain is made of, as well as the way the glycosidic linkages are arranged. So I'm going to say that again because it's pretty important. Biology is all about structure and function, how things are arranged and how they behave. So when we're talking about polysaccharides, their structure and their behavior, their function, depend on the monomers, the individual sugars that make up that polysaccharide chain, because there are plenty of different monomers, plenty of different monosaccharides out there. And it also depends on how the bonds come together, like how they're arranged in space. All right, so that may seem a little confusing, but just keep going with me. Let's get into this. We'll start with the storage polysaccharides. Many organisms, especially plants and animals, use sugar as a primary source of energy. Now, plants make the sugar that they use. Animals, including us, people, can't make sugar. We have to bring it into our bodies by eating plants or other animals. But either way, whether we're talking about a plant or an animal, when we're using sugar for energy, we're usually using it in monosaccharide form. What I mean by that is we're usually using individual sugar molecules. But plants typically make more sugar than they immediately need, and animals typically consume more sugar than they immediately need. So what happens to that excess sugar? Is it just wasted? Definitely not. Nature doesn't like to waste anything. The sugar molecules that are not immediately used are stored as polysaccharides. So let me say that again. So when we're using the sugar for energy, we usually use it in monosaccharide form. We usually break down individual sugar molecules. But the sugar that we're not using immediately and that we want to store for later, we're going to store it in polysaccharide form. We're going to store it in those long chains like we saw earlier. Now, I want you to think about medicine, like pills. So they typically come with, 30, 60, or 100 or more pills in one bottle. But let's say you have a fever and you want to take some pills. You'll probably take just 1 or 2 pills at a time, hopefully. You wouldn't take all the pills that are in the bottle. But you do want to keep the rest of the pills, though, because you could have a fever again in the future. So even though you take only 1 or 2 pills at a time when you're sick, it wouldn't really be efficient to store the pills in groups of just 1 or 2. It makes more sense to store them in large quantities. So this is kind of how sugar works. When you're using it for energy, you use individual molecules. But when it's being stored for later use, it's stored in bulk. All right, so we're not going to go too deep into this little cartoon here, but this is just showing a little animal eating grass, and the sugar that is coming into the animal's body is being stored. The sugar that he's not immediately using is being stored in polysaccharide form. All right, so again, when we're using sugar for energy, we're going to use it in individual monomer form. When we're storing it, we're going to store it in polysaccharide form. Now, if that makes sense, then put a thumbs up in the comments below so that I know that we're all on the same page. Now, like I said, both plants and animals store sugar, but we store it in 2 different forms. Plants store sugar in the form of starch. Starch is a polysaccharide that's made of glucose molecules. Now, remember I said earlier that the function of a polysaccharide, or how it behaves, depends on the monomers that it's made of. Well, starch is made of glucose, and glucose is the monomer that's used most often for energy. It's the primary energy source for most organisms. So since starch is made exclusively of these little energy molecules, it would make sense that it just becomes this big long chain of stored energy. So plants have little starch granules within their cell. So this is a plant cell, and inside of that cell, these are actually granules or like little pockets of starch inside that cell. So pretty cool to see that. So when the plant needs to get some sugar for energy, then it can take, these starch granules and hydrolyze the polysaccharide. So when I say hydrolyze, I just mean break it down. So break down that polysaccharide into its individual glucose monomers. All right. Now, starch comes in different forms, because why not? Why not make this harder, right? But we're just going to talk briefly about 2 of those forms, which are amylose and amylopectin. Amylose is an unbranched chain, and that means that all the monomers are connected in a single line. So I've got this illustration here. Now, you do see some loops, but those aren't branches. This is still a single line. There's no, different branched line of sugar molecules coming out. Now, remember again, I said that the structure of a polysaccharide, or how it's arranged, depends on how those glycosidic linkages are arranged. Basically, it depends on what carbon on one monosaccharide is attached to what carbon on another. So when we're looking at amylose, The monosaccharides in amylose have 1 to 4 linkages. What does that mean? That means that the number 1 carbon on one glucose molecule is attached to the number 4 carbon on the next one. So let's look at this a little deeper. So here is a glucose molecule, all right? So, and I've said this in a couple of videos, but when we're looking at a structure, any type of really organic structure, a lot of times you won't see the letter C to designate carbon. It's understood that the corners of the structures, like here, are carbons. So the carbons in organic structures are always numbered. I guess one person just started numbering them one way and everyone just kind of followed suit. So I'm going to go ahead and put numbers here. So this in this corner is carbon 1. Here's 2, 3, 4 in this corner, 5. And then the 6th carbon is this one up here that's outside of the ring. All right, so here in my second glucose molecule, I'm going to do the same thing. Okay. So now I've got both of my glucose molecules numbered. So what do I mean by that 1 to 4 linkage? This carbon 1 is going to bond with this carbon 4. So that's how these two glucose monomers are going to come together. The carbon 1 on one glucose monomer is going to join with the carbon 4 on the next. So that's a 1 to 4 linkage. That's all that means. Now, if you're in kind of a more general biology class or intro biology, you most likely don't need to know about the glycosidic linkages. This is probably more for AP or college level biology. Okay, so if we come to this illustration here, again, we see this 1 to 4 linkage. So this glucose monomer is joined to this one because of the bond closest to the carbon 1 here and the carbon 4. And I should clarify to say it's not the actual carbons that are actually bonding with each other. As we can see, there's an oxygen in between, but we still reference the carbon 1 and the carbon 4 because the oxygen is in between the carbon 1 here on this monomer and the carbon 4 on this monomer. Okay, so we've got that 1 to 4 linkage there, and that's just going to keep happening. So more glucose monomers are going to join. So here, this carbon 1 is going to get another glucose monomer, and it's going to bond here the carbon 4, and so on and so forth, until eventually we're going to end up with a long amylose starch chain. So again, this is a plant sugar, and this is one way that plants store sugar for energy in the form of amylose. Now, amylopectin is another plant sugar, another starch, and it's also made exclusively of glucose monomers, and it has the same function as amylose, which is storing energy. Now, amylopectin is different from amylose because of its structure. They have two different structures. So the glucose monomers here have 1 to 4 linkages, but they also have 1 to 6 linkages. So if we look over here, we see what we saw with the amylose. And here in this illustration, the carbons are numbered 4, so that's great. So here you see carbon 1 and then carbon 4. So that's where this bond is happening, this oxygen there between carbon 1 and carbon 4. Same here, carbon 1 and carbon 4. But then here we see something different. This carbon 1 is actually bonding to the oxygen that's near the carbon 6, the 6th carbon of this particular glucose monomer. Okay, and then we go back to seeing 1 to 4 linkages. So amylopectin has 1 to 4 linkages, and it also has 1 to 6 linkages. Again, this is more for kind of AP and college biology. Now, this makes things a little complex. It causes the amylopectin chain to be branched. So that's what we're seeing here. Amylopectin has a more complex structure than amylose, even though they have the same function. They're both used to store energy. So you can remember that amylopectin is the more complex starch because its name is more complex. Amylopectin is, a more complex name than amylose. So that's starch. What are some plant starches that we should be familiar with? We've got cornstarch, wheat starch, potato starch. These are all things that people and animals eat. Now, when we eat these plants, those starch chains enter into our digestive system, and we have enzymes in our digestive system that are going to hydrolyze them or break them down into single glucose monomers that we can use for energy. But like I said, we usually don't need all of the energy that we consume at that immediate time. So we're going to store it, but not as starch. Now, animals store their sugar in the form of glycogen. So glycogen is to animals what starch is to plants. Now, if I as an animal was to eat another animal like chicken or beef, then I would get glycogen from that animal, not starch. Okay, so let's take a look at glycogen. It consists of glucose, just like amylose and amylopectin. I just want to clarify that there are plenty of different monosaccharides out there, not just glucose, but glucose is, again, that primary energy monomer. So it makes sense that all of these different energy storage polysaccharides that we're looking at are all made of glucose. Now, glycogen is the most branched of the three storage polysaccharides that we looked at. So here's just an illustration of it here. You see that it's helical for one, which gives it that kind of curvy, loopy structure, but it's also really branched, okay, more so than amylopectin. Okay, so if you are in some type of higher level biology class where you may be expected to know the structural differences among these three storage polysaccharides, just look at the branching and you'll be able to tell them apart. Again, amylose has no branching. Even if you see these little loops, those aren't branches. Amylopectin is going to be somewhat branched, okay? And then glycogen is going to be very much branched. So for humans and other animals, the excess sugar that we take in is eventually pushed out of our bloodstream and into our liver cells and our muscle cells. So here you see a diagram showing that glucose pathway once it enters into an animal's body. And we're not going to get too deep with this illustration, but I do just want to highlight here you see in the liver, you see glucose turning into glycogen. So again, the excess glucose that we took in that we don't immediately need is pushed out of our bloodstream. It goes into our liver. That's one of the places where it goes, and it becomes glycogen, which is that stored polysaccharide. So here's actually an image. It can also be stored in our muscle fiber. So here is an actual image of a human muscle fiber. And these little dots here, if you can see them, are granules of glycogen. Okay. So we saw earlier, we looked at a plant cell and we saw those granules of starch. So here we're seeing the animal equivalent, the little granules of glycogen polysaccharides. Now, when we need energy, that glycogen is pushed out of our cells back into our bloodstream in the form of glucose, where it can flow to other parts of the body. Now, carbohydrates provide short-term energy, and polysaccharides can be stored for only about a day. So that means that carbs that you take in today would be gone by around the same time tomorrow. You would have to consume more carbs to get that energy. Therefore, taking in a very low amount of carbs can make you feel tired and weak. Now, there is another macromolecule called lipids, and it also provides energy, but it takes a while for that type of energy to kick in. So I'll do another video about lipids where I'll talk more about that. But carbohydrates are our first resort for energy because they break down quickly. But again, we store only about a day's worth of carbohydrates at a time. So let's go back to this diagram that we looked at earlier to kind of summarize what we just talked about. So this, again, we're talking about polysaccharides, which is the polymer of carbohydrates. So we said they have two functions, and we just talked about that storage function. So polysaccharides can be used to store sugar for up to about a day's worth of energy. And plants and animals store polysaccharides. So with animals, we talked about how sugar can be stored in the muscle and liver cells, and that is in the form of glycogen. That is a polysaccharide that is very extensively branched. Okay. Plants will also store sugar in the form of starch, typically in their roots and their seeds. Now, starch comes in two forms. We have amylose, again, which is the unbranched polysaccharide, and amylopectin, which is somewhat branched. I notice glycogen has just one form, it's glycogen. So glycogen and starch are equivalent, okay? Even though I don't have them on the same line here, glycogen and starch are equivalent. Starch just breaks down further into two different types, amylose and amylopectin. Now let's look at structural polysaccharides. These type of carbs are used as sturdy building material. For instance, every single plant has a wall around each of its cells, and those are called cell walls. And they use a polysaccharide that's called cellulose to build those cell walls. Also, arthropods like insects and crustaceans use a different type of polysaccharide called chitin to build their exoskeletons. Now let's take a look at cellulose to start. Just like the other polysaccharides that we looked at, cellulose is made of glucose. So wait a minute. If this is made of the same monomer that the other polysaccharides, the storage polysaccharides, are made of, then how come cellulose is used for building and not for storage? Well, remember, the structure and function of a polysaccharide depend on two things, the monomers and the glycosidic linkages. Now, in this case, the monomers are the same. Cellulose uses glucose, just like amylose, amylopectin, and glycogen. But the linkages are different. Now, glucose, that monosaccharide, actually has two different forms, alpha and beta. Now, I'm going to do a whole separate short video where I go into detail about the alpha and beta forms and how they're different. That's definitely for AP Bio, College Bio, but it is interesting, so everyone should check it out. But to kind of keep it a little simpler for our purposes here, cellulose uses what's called the beta form of glucose, whereas those storage polysaccharides, amylopectin, amylose, and glycogen use the alpha form of glucose. So this changes the whole structure of cellulose. It's straight. It's not helical like the others. So as we were seeing, especially with the amylose and the glycogen pictures that I showed earlier, you saw that they were helical. They were kind of like winding around. But cellulose, as we can see here, is straight. It's also never branched. Cellulose doesn't form any branches like how we saw with amylopectin and glycogen. Something else unique about cellulose is that it can form hydrogen bonds with other cellulose chains that are nearby. Now, why are these traits important? Let's think about building material. Now, the best building material is not going to be, flimsy like straw, or it's not going to be branched where you kind of have little pieces going everywhere like sticks. We want building material that's sturdy and can be straight and somewhat smooth, like brick. We also want material that can stick to other materials, like what happens when we put cement in between bricks. It makes it stick. The storage polysaccharides that we looked at would not make good building material because, as we can see, they're coily, they're helical, or they're branched, and they don't bond with each other. Like one amylose chain is not going to hydrogen bond with the second amylose chain. Cellulose, on the other hand, is like a smooth cable that can form a network, and that network is called microfibrils. So we see that here. It looks like yarn or string or some type of cable. Those are cellulose polysaccharides. So this is what helps build a plant's cell wall. And this is also how paper is made. Now let's think again about good building material. Do we want material that can break apart easily? No, of course not. We want material that would be able to stay together even if it's exposed to harsh elements. This is the same for polysaccharides that are used as building material. Now remember I said that when animals eat plants and get that starch, we have enzymes in our bodies to break that starch down. Again, not going to go too far in depth with this illustration here, but these scissors are kind of, they're of course pretend scissors, but they're showing enzymes that break down those starch chains into the individual glucose molecules that we see. So we can do that with starch, and that's what we want to happen. We want to be able to break the starch down because we want to get the energy out of it. So even plants need to break down the starch molecules that they build to harness the energy. But what plants don't want to happen is for their cell walls to break down, because that would endanger their cells. Those walls protect the cells. Well, here is what I would say is a beautiful example of intelligent design. The enzymes in our bodies that break down that starch actually cannot break down cellulose. They can't get past those beta-glucose linkages that I mentioned earlier. So most organisms actually cannot break cellulose down naturally. And this is great news for plants. And when we eat foods that contain cellulose, the cellulose is going to pass straight through our digestive system without being hydrolyzed, and it comes out in our poop. Now, there are a few organisms that can break cellulose down. Cows can't, but they have bacteria in their tummies that can break it down. So when cows eat grass, the bacteria in their stomachs break the cellulose in the grass down into glucose, which actually gives energy to the cow. Now, even though humans can't break cellulose down, it's still beneficial to us. So, for example, here are some foods that contain cellulose. So if we were to eat foods like this or anything with cellulose in it, and that cellulose gets into our digestive tract, it stimulates the tract to secrete mucus. And that mucus helps push the food that we're eating through our intestines so it can get through our entire digestive system. So that type of cellulose is known as insoluble fiber, and it's part of a healthy diet. When we're low in insoluble fibers, food can have a harder time passing through our digestive system, and that can lead to constipation, bloating, and a tough time on the toilet. Okay, so let's go back to our diagram, and this time we'll look at the structural or the building side of polysaccharides. So polysaccharides can be used as building material for cells and organisms. So we talked primarily about plants. So for plants, that type of polysaccharide is known as cellulose, and that forms unbranched fibrils, and that helps build the cell wall. So here's just an illustration here, again, very straight, and makes a nice little network that can be used to build that cell wall. I also mentioned arthropods, and fungi too also use a different type of building polysaccharide called chitin, and that's what helps build their exoskeleton. All right, and then you also have bacteria that use a third type of building polysaccharide called peptoglycan to build their cell wall. And that is all about carbohydrates. And carbohydrates are the quick energy macromolecule, the building macromolecule. Again, be sure to check out part 1 of my carbohydrate video. Well, that's it for today's video. I hope it was helpful for you. If so, don't forget to put a thumbs up in the comments section below. And if you have any remaining questions, please type those in the comments section and I'll do my best to provide you with an answer. Thanks again so much for watching CCL Biology, that is Christ-Centered Life. God bless and bye-bye.